Hi, welcome to Moreauknows. Today we're going to be learning about how a cell will maintain homeostasis in different environments. And the first thing you're going to need to know or to watch is the part 1 for this video, and that's on the plasma membrane. So for today our essential question is going to be, how is homeostasis maintained in a cell? Well, what does homeostasis mean? Homeostasis is going to mean that things will stay the same. Okay? And if things stay the same or are the same inside and out of the cell will be at homeostasis. So we're trying to keep a balance. What happens when food coloring is dropped into a beaker of water? Well, the molecules will spread and space themselves out far enough and evenly enough so that they will take up the entire volume of the beaker. And this is a basis of diffusion. We are moving from a high concentration of that one drop inside the beaker to a lower concentration where there are no food color molecules. And we can say the same thing if this was salt or sugar or anything like that. So if we had a chamber that was divided by a wall, and one side of the wall had a really bad smelling gas, and the other side did not. Well, if we were to open that door or remove that wall, all of the molecules would space themselves out evenly throughout the room. Okay, and this is going to be one part of diffusion. If we have a large concentration of sugar molecules on one side, let's call these glucose, on one side of the plasma membrane, then we will have a high concentration on one side. And then because there's a lot of sugar, and on this side there will be less sugar. So this would be a low concentration. Okay? And the molecules are going to want to flow to this lower concentration side until they are equal, meaning that they're flowing either way the same. And so here's the animation that you can see this happening. They're going to flow, flow, and flow until that net flow becomes so little and the flow either side is going to become equal. And now they are both equal on either side. So what does this mean for concentration? Well, if I was taking this would be a high concentration and this side would be a low concentration. And if I wanted to see where these molecules will move, well, they'll always move from a high concentration, and I like to think of this as a slide, to a low concentration. And so you can draw a little ladder if you want, and imagine kids playing on it, but it doesn't take any energy to move down the slide. So this diffusion does not take any energy, it happens naturally. So what are the different types of movement through a cell? Well, these are all going to be called transport, but we have the passive transport, which is that high to low, or moving in this direction. If we had a high to low, moving in the opposite direction from low to high, this would be considered active. So in passive diffusion, we can see that there's just regular diffusion. And passive diffusion means that it's not going to take any energy or any proteins that will need to assist in the movement of molecules. And so we can use diffusion, passive diffusion, as for things like oxygen and carbon dioxide, and even osmosis, which is the transport of water. And we'll get into that in just a second. The facilitated diffusion needs proteins to help with the transport. So it facilitates the movement through the plasma membrane, but it still does not take any energy. So if it's in passive, it's no energy. Okay? And if it's active transport, then it's going to need energy. Sorry for my horrible handwriting. the reason it's going to need energy is because the molecules are going to be pushed against their concentration gradient. Okay? So, in active, we need energy. So we're going to need some proteins to actually do the pumping that's needed to get these molecules across. So we need some sort of pump. And the energy currency will be in the form of ATP. And the most famous one is for energy is the sodium, Na, and potassium, K, pump. And this protein will actually pump these across the molecule. And we're also going to need energy for endo and exocytosis. Endo means that that molecules will move inside the cell, enter, endo, and exit, exocytosis. So this is for molecules moving inside and outside of the cell. The first thing is going to be passive transport. So passive transport can happen in two ways. We have just regular diffusion, which is molecules like oxygen and carbon dioxide just being able to move through the plasma membrane without any problem. And then sometimes with water and with other molecules that are maybe charged or something like that, need a protein to kind of go through. And so these molecules will actually move through the protein and be able to diffuse without using any energy. Some of these molecules will actually have to open up and then let one molecule through at a time. And so the molecule will kind of open on one side, close on the other, and keep pumping it through like that. But again, not using any energy. It's passive. Just like if you were sitting there right now not taking your notes, you would be passively sitting there listening to me speak. But if you were actively listening to me speak, you'd be using energy, using your brain, writing things down, and that would make you active instead of passive. Osmosis is the movement of water from a high concentration of water to a low concentration of water. So it's the same thing as diffusion, going down this concentration gradient without using any energy. And so how do I know which side has more water or less water? Well, if it's a hypertonic solution, it's going to have less water in it. And that is because there are more molecules. So for every molecule that we have on one side of this plasma membrane right here, there's going to be less water. And there's going to be more water over here because they're at the same volume. Okay? So if I put a bunch of rocks into a beaker and a small amount of rocks into a beaker and I filled them up to the same amount, which one would have more water in it? Well, this one would have more water in it. And this is a hypotonic. So you can think of hypo being like a hippo. So hippo, think more water. Hyper, you drain yourself. Okay, you're running around all hyper and you're all sugared up, and then usually you pass out. So think of hyper being less. Okay, water will always flow from a high concentration to a low concentration. And so that's what we see here. The water will actually, if we let this go and there was a membrane right here, the water movement would actually move into this side of the cell to reach an equilibrium because it wants to be evenly keeled. And so now, because the water has moved over here and there's enough concentration of solutes versus water, this is actually at a isotonic or equal solution. And so we can name these things. If a molecule is in a hypertonic solution, the water is going to move out of the cell. Okay? And so hypertonic means that it has more molecule or more solute outside of the cell. And so water is going to move in. And on the opposite end, if there were more solutes inside of the cell, and this was the in a hypotonic solution, then water's going to move into the cell, making it really big or even maybe explode. And if the balance is just right, meaning the solute concentration is equal, and so there's no high or low, then those will actually be at what's called isotonic. And water can move freely in and out. It doesn't stop moving, but it can move inside and out. Water can move inside and out freely. And so to kind of test this, if I have more solutes outside of a slug, let's say I have a bunch of salt, and I put that onto a slug, and I put all this salt on, what will happen? Well, there's more solutes on the outside of the cell, and all of the cells in the slug will move out, causing the cells in the slug to shrivel. And this is very cruel, and you should never ever do this, but they will, because it will kill the slug. Now let's talk about active. So most of you guys are sitting there taking notes, right? You're actively moving around and listening to me and taking notes. So active transport is going to use energy. And so right here is a sodium-potassium pump, which is kind of the ones that they always use in the EOCs or any tests that they have for biology. And so sodium will actually move in one direction, and potassium will move in the other direction. And they will both be going across or against the concentration gradient. So usually we have a high to low situation where things kind of move down like a slide or a waterfall. Well, if you had to run up that slide, you're going to need a lot of energy. And so that's what I think of when I use active transport. Or if you were flowing uphill, if you were a kayaker, you'd be flowing up the river. Or if you had to ski going uphill, you're going to have to use a lot more energy than just skiing down the hill. Or snowboarding. I like snowboarding. So you use energy. So here we have regular diffusion, where we move from a high concentration to a low concentration. If we use facilitated diffusion, we're still moving from a high concentration to a low concentration. And in this facilitated diffusion, we can have two different types of protein, but that's not going to really be necessary for honors biology. And then the sodium-potassium pump, we're moving from a low concentration of this molecule to a high concentration inside. So it's actually moving in. So this molecule in the opposite motion will be going against the concentration gradient the other way. And every time it clicks back and forth. Our last two topics are endocytosis and exocytosis. So when I think of endo, I think of enter. So I've got the N and the N. So that means to enter the cell. And so this plasma membrane will actually make a kind of movement around a section that it wants to bring into the cell. And it will actually close this off and bring this vesicle into the cell. And that's endocytosis. And it takes some energy, so it's in the active transport. Exocytosis, so when I think exo, I think exit. And if I'm exiting someplace, I'm leaving that place. And so the X and the X make sense. So if I had a vesicle that wanted to leave the place, maybe it was coming from the Golgi body, which was transporting some proteins to go to another cell. Well, these proteins inside this vesicle need to get out. And so the way that it does that, it will slowly be absorbed into the plasma membrane of the cell, and then eventually start to open up into maybe like a little hole, and then that hole will start to get bigger as these ends will pull in either side, and then eventually this will force all of the proteins out, and then afterwards this will move up, and that same plasma membrane from the vesicle will become part of the plasma membrane on the outside, because remember, it's a fluid mosaic model. And so it won't even be a vesicle anymore. It'll just become part of the cell. And I always thought that was kind of cool because it just kind of bubbles in and bubbles out. Thank you for watching Moreauknows. Now you do.